Welcome my dear students in today's session. Today we are going to discuss about a common tropical protozoal disease in our country, that is Leishmaniasis. Leishmania. Leishmania is a hemoflagellate. We know flagellates are of three types: intestinal flagellate, vaginal flagellates and hemoflagellates. Hemoflagellates are of two types, that they are Leishmania and Trypanosoma. Leishmaniasis is caused by Leishmania species. Clinically, Leishmaniasis is manifested as visceral leishmaniasis, cutaneous leishmaniasis and mucocutaneous leishmaniasis. Genus Leishmania and medically important species of Leishmania are Leishmania donovani, Leishmania tropica, Leishmania brasiliensis, Leishmania mexicana, Leishmania ethiopia and Leishmania jagassii. Risk factor of Leishmaniasis. Children and males are more affected, and low socio-economic status is also responsible for Leishmaniasis. There is a seasonal variation, high prevalence during the pre and post monsoon seasons. The disease is usually confined to rural area because of more breeding place for sandflies. Clinical classification of Leishmaniasis. Number 1, visceral leishmaniasis or kala jar. Kala jar is caused primarily by Leishmania donovani, Leishmania jagassii and Leishmania infantum. Cutaneous leishmaniasis. Cutaneous leishmaniasis is caused by Leishmania tropica, Leishmania mexicana, Leishmania ethiopia and Leishmania major. Number 3, mucocutaneous leishmaniasis, it is caused by Leishmania brasiliensis. Cutaneous leishmaniasis is again classified into three groups. 1, dermal leishmaniasis or PKDL, that is post kala jar dermal leishmaniasis. It is a sequel of visceral leishmaniasis. Number 2, old world leishmaniasis or old world cutaneous leishmaniasis. It is caused by Leishmania tropica, Leishmania ethiopia, Leishmania major. Number 3, new world cutaneous leishmaniasis. It is caused by Leishmania mexicana, Leishmania guanensis and Leishmania brasiliensis. Now, morphology of Leishmania. All species of Leishmania exist in two morphological forms. Number 1, amastigote forms and number 2, promastigote forms. Number 1, amastigote. Amastigote is usually short, oval shaped and has no flagellum. It is non-motile and intracellular. It resides in reticuloendothelial system, that means mononuclear cells. Mononuclear cells are monocyte, macrophage in lymph node, spleen and lung, Kupffer cell in sinusoid of liver, microglial cells in the central nervous system and dendritic cell. An important structure of amastigote and promastigote forms is kinetoplast. Now, what is kinetoplast? Kinetoplast is a DNA containing rod shaped structure that lies near the anterior end of amastigote and promastigote form. Nucleus is situated in the middle of the cell or side of the cell wall. Amastigote form is also called the Leishman-Donovan body or LD body. The figure shows different forms of hemoflagellates. Amastigote and promastigote forms are seen in Leishmaniasis, and Trypanosoma has two morphological forms, that are epimastigote and trypomastigote. Here is a picture of amastigote form. Now, promastigote. The stage of parasite is found in the cultures and insect vectors, that means sandflies. This is the infective form for man. The kinetoplast is towards the anterior end and a free flagellum with no undulating membrane is emerged. Here is a picture of promastigote. Now, vector. Leishmaniasis is a vector-borne parasitic disease. It is transmitted by female sandflies of genus Lutzomia and Phlebotomus. In New World Leishmaniasis, the vector is Lutzomia species, and in Old World Leishmaniasis, the vector is Phlebotomus species. Here is a picture of sandfly, which is very common in Bangladesh. Especially Leishmaniasis is endemic in Mymensingh, Netrokona, Kishoreganj, Tangail, etc. Mode of transmission: by the bite of female sandfly, the disease is transmitted. Direct transmission may also occur accidentally by inoculation of parasite during laboratory work or by blood transfusion, and congenital transmission may also occur, but rarely the fetus can affected by placental defects. Reservoir host: depending on the geographical area, the reservoir can be either a wild or domestic mammals such as fox, jackal and rodents, or even in the particular cases human beings. Important events of life cycle: definitive host human, jackal, rodent, dog. Intermediate host sandfly. Site of localization mononuclear phagocytic system. Infective form promastigote, and diagnostic form is amastigote in the tissue macrophage and promastigote form in culture. Now, we are going to discuss about the life cycle of Leishmaniasis. At first, promastigote form enters into the human body by bite of female sandfly from the pharynx of the sandfly. It is injected to host by blood meal, and promastigote form is ingested by macrophage. After going into macrophage, it changes into amastigote form. Amastigote form multiply by binary fission. It can traverse through macrophage in different body tissue. Amastigote in skin tissue can be picked up again by sandfly by taking blood meal. Then amastigote form is moved to midgut or hindgut of the sandfly and converted into promastigote. Promastigote form then multiply and goes to the salivary gland or pharyngeal area of the sandfly. Then again promastigote can be entered into human body or new host by taking another blood meal. Thus, the life cycle is continuous. Amastigote form within macrophage reside within reticuloendothelial system and thereafter undergoes multiplication. When the parasitized macrophage goes to circulation as wandering macrophage, it leads to a generalized infection. It causes reduced bone marrow activity or bone marrow hypoplasia, hepatosplenomegaly and lymphadenopathy. Pancytopenia can occur due to formation of autoantibody against red blood cell, white blood cell and platelets. That is all for today's session. In next class, we will discuss about the pathology, pathogenesis and laboratory diagnosis of Leishmaniasis. Thanks a lot for your patience hearing.